Welcome to another episode from the Veterinary Medicine Digest. Imagine for a moment a small, folded structure tucked neatly at the base of the brain, a structure that for centuries was dismissed as a mere assistant to the grander cerebral cortex. For much of our history, we believed the cerebellum was simply a biological metronome, a silent regulator of movement and balance that kept the body from stumbling over itself. But as we peel back the layers of neurological mystery, we find a story that is far more complex and far more scandalous in its implications for how we understand the canine mind. We are discovering that this little brain is not just a motor coordinator, but a master of prediction, an arbiter of emotion, and a guardian of cognitive focus. Today, we are going deep into the heart of canine neurology to explore the hidden geography of the cerebellum and the groundbreaking research that is finally allowing us to map specific clinical syndromes to the very lobules and nuclei that define them. Our journey begins in the late 18th century with a scientist named Malacharn, who was among the first to truly look at the cerebellum and provide names for the features we still recognize today, such as the vermis and the pyramid. Malacharn even theorized that the number of folds in this structure might be tied to the level of intelligence in a creature. Decades later, a pioneer named Flourens demonstrated through his work with pigeons that damaging this area led to a catastrophic loss of coordination, a finding that cemented the cerebellum's reputation as a purely motor organ for over a century. We saw a triad of symptoms emerge in these early animal studies: atonia, asthenia, and astasia, which described a world of decreased resistance, movement weakness, and involuntary oscillations. But as the 20th century came to a close, a new era of research, led by figures like Jeremy Schmahmann, revealed that the cerebellum was communicating with nearly every region of the cerebral cortex. In humans, this led to the discovery of the cerebellar cognitive affective syndrome, or Schmahmann syndrome, where damage to the posterior regions of the cerebellum resulted in profound changes to executive function, language, and emotional regulation. In the world of canine medicine, we have long struggled to match this level of anatomical precision. We knew that a dog with cerebellar disease might present with a wide-based stance, intention tremors, or a lack of a menace response, but pinpointing the exact lesion based on those signs remained an elusive goal. That is, until now, a massive multi-center retrospective study involving over 100 client-owned dogs across Spain and the United Kingdom has finally provided us with a definitive correlation between cerebellar lesion topography and the differential diagnosis of clinical presentations. This study has identified 3 distinct region-related cerebellar syndromes in dogs that parallel those found in humans: a motor syndrome localized to the rostral lobe, a behavioral syndrome found in the caudal lobe, and a vestibular syndrome tied to the deep white matter and the interpositus nucleus. To understand how we reached these conclusions, we must first look at the basic structural anatomy that serves as the stage for this neurological drama. The cerebellum is a highly conserved organ across mammals, organized into 10 distinct lobules numbered 1 through 10. It contains more than 60% of all neurons in the brain, despite occupying only 10% of the brain's total volume. The cerebellar cortex is an exquisite machine of calculation, arranged into 3 layers: the molecular layer, the Purkinje cell layer, and the granular layer. The Purkinje cells are the stars of this show, acting as the sole output of the cerebellar cortex, sending inhibitory signals to the deep cerebellar nuclei, which then project outward to the rest of the nervous system. These circuits are organized into longitudinal modules that process information in parallel loops, acting as internal models that predict the dynamic properties of the body and the environment. When this predictive machine fails in a dog, the results are often sudden and dramatic. Let's look at the first of our identified syndromes, the cerebellar motor syndrome. In the study of 102 dogs, researchers found that lesions in the rostral cerebellar lobe were significantly associated with primary motor abnormalities. Specifically, damage to the rostral culmen was linked to cerebellar ataxia, a classic lack of coordination that leaves a dog staggering and unsure of its steps. Even more striking was the association between the lobulus centralis and a terrifying condition known as dicerebellate rigidity. A dog suffering from this will exhibit a dramatic opisthotonus, where the head is pulled back, the thoracic limbs are hypertonic and extended, and the hips are flexed in a posture of extreme neurological distress. This rostral lobe is essentially a pure sensorimotor structure, and when it is compromised, the dog loses the ability to regulate the speed, range, and force of its movements. But move your attention further back to the caudal lobe, and the story changes from one of coordination to one of character. The researchers discovered that involvement of the folium and tuber vermis was linked not to motor deficits, but to abnormal behavior. Dogs with lesions in these areas were more likely to be disoriented, subdued, lethargic, or hyperreactive. This is the canine equivalent of the cognitive affective syndrome, providing evidence that the caudal cerebellum is deeply involved in the limbic and emotional processing of the dog. It is a revelation that challenges the old guard of neurology. It tells us that if a dog's personality seems to vanish after a neurological event, the answer might not be in their cerebrum, but in the back of their head. Then there is the third syndrome, the one that bridges the gap between balance and the internal sense of space, the cerebellar vestibular syndrome. This is where the deep white matter of the cerebellum, particularly the area involving the interpositus nucleus, takes center stage. The study revealed that lesions in this deep region were significantly associated with a head tilt, nystagmus, vestibular ataxia, and delayed postural reactions. This area is a crucial node in the vestibulocerebellar loop, communicating directly with the vestibular nuclei in the brainstem. Damage here often leads to a paradoxical vestibular syndrome, where the dog tilts its head toward the side, opposite the lesion, because the normal inhibitory signals from the Purkinje cells have been lost, leading to an explosion of vestibular activity on the side of the damage. This mapping of syndromes is not just an academic exercise. It is a vital tool for differential diagnosis through what veterinarians call the vitamined mnemonic, which stands for vascular, idiopathic, traumatic, anomalous, metabolic, inflammatory, neoplastic, and degenerative. Each of these disease categories leaves its own unique footprint on the cerebellar landscape. For instance, the study found that vascular disease, such as an ischemic stroke, was significantly associated with brachycephalic breeds, such as the Cavalier, King Charles Spaniel, and the Pug, as well as dogs with pre-existing comorbidities. These strokes often had a peracute or acute onset, frequently leaving the dog non-ambulatory and suffering from vomiting. Interestingly, these vascular lesions were almost never diffuse. They were localized, wedge-shaped insults that favored the rostral vermis and the rostral paravermis. In contrast, neoplastic conditions, or cancers of the cerebellum, were found to occur in older dogs and were significantly linked to signs of incoordination and a persistent head tilt. Unlike strokes, these tumors showed no specific topographic preference, appearing wherever the biological dice happened to roll within the cerebellar structure. Then we have the inflammatory and infectious diseases, which often presented a starkly different clinical picture. These dogs frequently exhibited signs of pain and, most notably, tremors. This brings us to a specific condition known as idiopathic generalized tremor syndrome, formerly and somewhat controversially called little white shaker syndrome. The history of idiopathic generalized tremor syndrome is a classic example of how a misnomer can stall medical progress. For years, the condition was thought to only affect small white dogs, like the West Highland White Terrier or the Maltese. But a large multicenter study of 75 dogs with this syndrome showed that crossbreeds were actually the most commonly affected, and dogs of all coat colors could fall victim to these full-body tremors. This syndrome is believed to be an immune-mediated inflammation of the central nervous system, and while the name suggests a focus on the cerebellum, the tremors themselves and the presence of seizures in some cases suggest a more diffuse disease process. These dogs are typically young, often around 17 months of age, and they respond remarkably well to corticosteroids, although relapses are common. The inflammatory nature of cerebellar disease is being further illuminated by the search for neural autoantibodies. In humans, the discovery of antibodies that target the surface of neurons has revolutionized our understanding of autoimmune encephalitis. Now, we are seeing similar findings in dogs. A study looking for these markers in dogs with idiopathic generalized tremor syndrome found an increased titer of metabotropic glutamate receptor, 1 antibodies in a subset of patients. This receptor is critical for the long-term depression of synapses within the cerebellum, a process essential for motor learning. When these receptors are attacked by the dog's own immune system, the result is a severe cerebellar syndrome that may include hyperthermia and significant abnormalities in the cerebrospinal fluid. As we move from the acute insults of stroke and inflammation into the long-term challenges of degeneration, we find a different kind of tragedy. Hereditary ataxias in dogs are a large group of neurodegenerative diseases that share a core feature of cerebellar or spinocerebellar dysfunction. These are often caused by genetic variants that interfere with basic cellular functions like protein degradation or cation trafficking. One of the most heartbreaking forms is the primary Purkinje cell degeneration, or cerebellar abiotrophy, which has been identified in breeds ranging from the Beagle to the Finnish Hound. In these dogs, the Purkinje neurons, those master calculators of the brain, undergo a premature and spontaneous death. The signs can begin as early as 3 weeks of age, as seen in beagles, where the puppies frequently miss their food bowls and tumble over their own feet as they try to navigate a world that their brains can no longer predict. The study of these degenerative diseases has also led to the proposal of a new category, multifocal degenerations with a predominant spinocerebellar component. This includes canine multiple system degeneration, found in the Kerry Blue Terrier and the Chinese Crested, where the disease strikes not just the cerebellum, but also the extrapyramidal nuclei, like the substantia nigra. These dogs start with a mild tremor, but progress relentlessly to a state of akinesia, unable to stand, as their nervous systems slowly go dark. It is a stark reminder of the cerebellum's interconnectedness. It is not an island, but a hub in a vast network of biological communication. This brings us to the fascinating and often overlooked role of the cerebellum in autonomic functions, specifically micturition, or the act of urination. While we often think of the bladder as being under the control of the lower spinal cord or the cerebral cortex, research has shown that the cerebellum, particularly the fastigial nucleus and the vermis, plays a critical role in the micturition reflex. In experiments with decerebrate dogs, removing the cerebellum led to a decrease in the threshold volume required to trigger the bladder to empty, and a subsequent decrease in the pressure of the contraction. This suggests that the cerebellum acts as a tuner for the bladder, ensuring that the act of urination is as coordinated and efficient as a well-timed leap or a graceful trot. But what happens to this incredible organ as the years roll by? The cerebellum, like all parts of the body, is subject to the relentless march of aging. We see structural and functional changes that can lead to what is known as the physiocognitive decline syndrome. Studies using magnetic resonance imaging have shown a negative correlation between cerebellar volume and age, often mirrored by a decline in motor performance and processing speed. However, the brain is a resilient thing. We have discovered the concept of the cerebellar reserve, the remarkable capacity of the cerebellar circuitry to compensate for damage or loss. This reserve can be structural, where healthy areas take over for a damaged region, or functional, where the surviving neurons in a degenerating area work harder to maintain the predictive models of the body. This resilience is why some dogs can recover so well after a stroke. In a study of 23 dogs with rostral cerebellar ischemic strokes, every single dog survived and was discharged within 10 days. While they may have arrived at the hospital in a state of severe neurological collapse, their brains immediately began the process of remapping and self-repair. This is the scandal of the cerebellum. It is so much more than we gave it credit for. It is an organ of incredible plasticity, sensitive to environmental enrichment and training. There is even evidence that lifelong practice, such as musical training in humans, or complex agility tasks in dogs, can help maintain the integrity of the cerebellum into old age, protecting it from the atrophy that would otherwise steal its function. As we look to the future, we are seeing the rise of non-invasive brain stimulation techniques, like transcranial direct current stimulation, which are being used to modulate the excitability of the cerebellum, to treat everything from chronic pain to the tremors of Parkinson's disease. These techniques may one day allow us to boost the cerebellar reserve in aging dogs or help those recovering from a traumatic brain injury. We are also beginning to understand the intricate link between the cerebellum and the gut-brain axis, exploring how diet and the microbiome might influence neuroinflammatory disorders like idiopathic generalized tremor syndrome. But for all our progress, there are still vast territories of the cerebellum that remain unmapped. Our understanding of canine cognition is still in its infancy, and we have yet to develop standardized rating scales that can truly capture the subtle behavioral changes that occur when the caudal lobe is damaged. We are still working with presumptive diagnoses in many cases, lacking the histopathological confirmation that would provide the final piece of the puzzle. And we are still learning how to translate the incredible precision of optogenetic mapping in mice, where we can initiate or terminate movement with a flash of light into the clinical reality of the veterinary exam room. The story of the cerebellum is a story of a silent partner finally finding its voice. It is a story of 3 syndromes that allow us to look at a dog and see the specific lobules that are struggling to keep their world in focus. It is a motor coordinator, yes, but it is also a predictor of the future, a balancer of emotion, and a resilient guardian of the self. When we see a dog with a contralateral head tilt and ipsilateral postural deficits, we are no longer just seeing a mystery. We are seeing the deep white matter calling for help. When we see a dog with decerebellate rigidity, we are seeing the lobulus centralis in crisis. And when we see a dog who has become a stranger to their family, we are seeing the folium and tuber vermis, losing their grip on the dog's emotional world. In the end, the study of the canine cerebellum reminds us that the mind is a landscape, and every valley and peak has a purpose. It teaches us that even the smallest parts of our biology can hold the largest secrets of our lives. As we continue to refine our maps and sharpen our diagnostic tools, we move closer to a world where no dog has to suffer in a neurological fog that we cannot understand. We are finally learning to listen to what the little brain has been trying to tell us all along. Thank you for listening. The titles for the referenced papers are in the video description. From VetMed Digest, where we keep minds inspired, hearts light, and tails wagging.